Participants are joining now.
Yes.
So we'll start by exactly by 11.
Yes, that will be
But
nice.
if there is some time required to join the participants, then we will start 1 or 2 minutes late.
That's fine.
Yes, yes.
Yeah. I think they are coming up. We still have another 4 minutes.
Yeah.
Yeah,
6 minutes. So, what is the total strength of this college in totality?
Yeah, total strength 4,500.
Okay. All right.
Junior, senior as well. Actually, senior is 3,000.
Equal participation from girls and boys.
Yeah, actually, girls are rather more participation than boys.
That's nice to hear.
Yeah.
Yes, yes, yes. Good morning, Dr. Watkar.
Good morning, madam. Good morning.
Good morning.
Good morning, sir. Good morning.
So I understand you all are still online only, all teaching programs at the moment, or
Yes.
on
Yeah,
site.
yeah. Right now it is on the online mode.
Okay.
Good morning, Kurekar, madam.
Korekar madam. Yeah.
Good
Good
morning,
morning.
sir.
Good
Very
morning.
happy to see Jingde madam also. Ma'am, welcome to Sangli.
Thank you very much. Thank you, Dr. Jayanti. It's nice
Thank
to
you.
see you finally here.
Thank you, sir. Thank you so much, ma'am.
Dr. Jayanti has been instrumental in assisting in this
Yeah, yeah.
coordination with the college.
Thank you.
Thank you, ma'am. Thank you so much.
I hope that by reaching out to all of you, we would be able to help in whatever way we can with the science that we know and bring it
मॅडम या इथे आवाज का येईना गेले
आवाज येतोय मॅडम. We can hear you.
येतोय.
आम्हाला येतोय मॅडम आवाज.
ऑनलाईन म्हटलं की थोडं कनेक्टिव्हिटी रेंज ॲट द रुरल एरियाज हे सगळे प्रॉब्लेम आहेतच.
we realize and that's why we are all ready for backups and hoping that the lecture will go without hitches. Why even in Mumbai we have these problems in different areas, so it's not necessarily only in the south.
Yeah, Jingde ma'am, but we have lots of problems over here. The internet connectivity, Wi-Fi, everything has a lot of problems. But then we have been doing whatever we can with everything, all the problems.
And that's called facing the challenges. Yes. Yes. I think that's the way to go ahead, and we can break all barriers and reach out to each other in whatever way we can.
It's a wonderful thing that we can do that in this new age of technology. Everyone has been connected so fast and so, widely.
Yeah, it's amazing. I acknowledge that, and yes, it is. It's been an amazing experience, especially for senior citizens like us who had to learn a lot.
But actually all senior citizens and even the research students also get acquainted with this recent technology, online lectures, and even the people are not necessary to go for any LMS type of technology. People have been joined now.
Yeah.
Including us.
Oh, is that including us? We don't know how many are there on what
62.
YouTube.
62.
No, on YouTube, I think he's got.
YouTube, there may be a lot of, but I think on Google Meet the 62 are joined.
Yeah, we can check on what's happening on the YouTube also.
एक 10-15 मिनिटांनी.
We have 24 on the YouTube at the moment.
34 on the YouTube, yeah.
We have 24 on the YouTube.
So 34 and this, so about. Yeah.
You can start, sir.
Yeah, we can
Anushka,
start.
Yes, sir.
please take charge.
A very good morning to one and all present here. I am Anushka Roy and I am a third year student studying at Department of Biotechnology at Smt K.W.College College of Arts and Science, Sangli. A very warm welcome to all of you. It is a wonderful day and I feel privileged to have this opportunity to address today's event. The world is changing at a fast pace, and it is really important for all of us to stay updated and constantly look forward to gain knowledge. Hence, our today's, our today's event is a popular science lecture organized by Indian Women Scientists Association, Vashi, Navi Mumbai. It is supported by Board of Research in Nuclear Science, BRNS, in association with Department of Biotechnology, Smt Kasturbai College of Arts and Science, Sangli. The topic of today's webinar is the application of recombinant DNA technology in science and medicine. To present this webinar, I would like to welcome our today's guest of honor, Dr. Saurabh Dalal Sir, Scientist Officer H at ACTREC, Navi Mumbai. Now, I would like to welcome Dr. Surekha Zingre Ma'am, Trustee of Indian Women Scientists Association and Coordinator of BRNS Lectures. I would like to welcome our respected Principal, Professor Dr. B.P. Ladgaonkar Sir, Dr. Gore Sir, NAP Coordinator, Dr. Hanmane Sir, our Vice Principal of Science Wing, Professor Dr. Jaya Kurhekar Ma'am, and Professor Dr. M.D. Vadmare Sir, Head Department of Botany, Professor Dr. S.P. Nikhalji Sir, Head Department of Zoology, Dr. U.M. Yadav Ma'am, Head Department of Microbiology, and Dr. S.S. Wadkar Sir, Head Department of Biotechnology. A very warm welcome to all the teachers and all my fellow friends. May I now request Professor Dr. B.P. Ladgaonkar Sir to welcome our esteemed audience. Sir, over to you.
Okay, thank you, madam. May I audible?
Yes, sir.
Yes, yes.
Yeah. Good morning, one and all. I myself, Dr. B.P. Ladgaonkar, Principal of this college, Smt K.W.College College of Arts and Science, Sangli. Indian Women Scientists Association, Navi Mumbai, and Department of Biotechnology of Smt K.W.College College of Arts and Science, Sangli has organized this webinar. For this webinar, the speaker is Dr. Saurabh Dalal. He is going to talk about the applications of recombinant DNA technology in science and medicine. I welcome him on this online platform. Then from IWSA, Surekha Jingde Madam, I also welcome her. Then Dr. Jaya Kurhekar, I also welcome her in this online platform. In addition to this, the dignitaries of Indian Women Scientists Association, IWSA, the trustees of IWSA, the members of Executive Council of IWSA, members of IWSA from headquarters and all other branches of this IWSA, I, as a principal of this college, welcome you all in this webinar. Yes? Yeah. So, the vice principals of this college, Dr. P.N. Gore sir, Dr. Hanmane sir, Dr. P.N. Chaugle sir, heads of all departments, particularly departments of biotechnology, department of chemistry, department of botany, department of zoology, and the different departments of life science together. This webinar is organizing. All faculty members of this college, teaching as well as non-teaching faculty members of this college, and all delegates, scientists, participants who are assembled over here on this online platform for this webinar, both Google Meet as well as YouTube link. I welcome you all. Dear friends, Kasturba Vachan College is one of the renowned college in the Shivaji University jurisdiction. The college is having adequate infrastructure right from junior college, senior college, and even in the PG program. The college is imparting quality education to the about 4,500 students among the different disciplines. The college is having about 22 departments from arts faculty and the science faculties, and major of the departments are ICT based infrastructure, and they are availing this infrastructure for learning and teaching and learning processes. The college is having MSc courses, MSc in physics, MSc in chemistry, MSc in particularly in electronics, MSc microbiology, MSc mathematics, and it gives me a great pleasure to say over here that we have introduced right from this year that one innovative course, that MSc in embedded technology. In addition to the PG programs in science faculty, there are PG programs in arts faculty also. That is MA English, MA History, MA Political Science, and so on. There are 8 different programs at the MA level, that postgraduate level on the arts faculty. Then at the onset, I would like to congratulate the Department of Biotechnology and all the Department of Life Science that they have organized this type of the webinar, particularly for the students and the faculty members, and they are doing rather pioneering job in imparting quality education to the students of this discipline. Now coming to this webinar, friends, with the major objectives, it is proposed to expose the students and the faculty members of the to the recent trends and the state-of-art technologies of this discipline. Particularly, this webinar is organized. This webinar is organized in collaboration with the Indian Women Scientists Association. Basically, this is running with the Department of I think Atomic Energy, that is Board of Research and Nuclear Sciences. This board is funding different types of projects in the different types of areas of the physics, areas of the electronics, areas of the chemistry, and all. And even such type of webinar also supported by the BRNS department of the Department of Atomic Energy. So before proceeding further, I would like to thanks to the authorities of IWSA, the trustees of IWSA, and the executive councils and other members of the IWSA that who has accepted our request and sanctioned this type of the lecture among the lecture series, popular science lecture series to this college. I am most grateful to the entire team of IWSA. Once again, I welcome you all and wish you best for this webinar and whatever the deliberation taking place during this webinar. So thank you, madam. Thanks. Over to you, madam.
Thank you, sir, for your guidance. Now, moving further, I would like to invite Professor Dr. Surekha Dhingre ma'am, trustee of IWSA and coordinator of BRNS lectures, to give an introductory speech about the popular science lecture series and about IWSA activities. Ma'am, over to you.
Thank you, Anushka. That was nice of you. Dr. Ladgaonkar, principal of the college, all my colleagues on this dais who are the heads of departments of microbiology, botany, zoology, Dr. Jaya Kurekar, and my colleagues from IWSA. I'm very happy to be here with you today on this lecture. On behalf of the trustees of IWSA, the executive committee members, and the members from the headquarters and branches, we extend a warm welcome to each and everyone who's present today, and in particular the youngsters who are here to anxiously hear the lecture. And before we go ahead, you must have a little glimpse of what we are at IWSA and see what we do to ensure that we make all our efforts to take science to society. And in a very short, quick presentation, I will show you what we do, and I hope you will know and come to know what IWSA is all about. So as I mentioned, can you see this presentation? Can you please confirm that? Yeah. So this is our headquarters based in Vashi. We are a non-governmental organization, secular, non-political, social welfare organization which believes in working for the people. And we are associated and registered with the Niti Aayog with a unique ID. And this, we are entering our 50th year, and what best time to look back to what we have done, to guide us to what we should be moving ahead to, and to do much better than what we have done before. So all the young ones, remember that this is the way we should be moving ahead all the time, to take on the best that we have learned from our past and what we will take forward to help us on our movements ahead. So for IWSA is in its 50th year. We complete 50 years on June 13th, 1923. And as I mentioned, the headquarters in Vashi. We have 11 branches across the country, Roorkee in the north, with Nellore in the south. They were all open at different parts, different times in the years. We have about 2,100 IWSA members. Our main mandates are to take science to society, and through that also for women empowerment. We work in two arms, community welfare and science awareness. And IWSA in 2019-20 reached out to about 23,000 recipients. And in 2020, though the pandemic was raging, we still could reach out to about 17,000 recipients. And that is thanks to the digital technologies which were available. Among the community welfare activities, we have the Working Women's Hostel, which was started way back in 1989, a healthcare center, a computer center, a library, scholarship awards, and early childhood care education program. The Working Women's Hostel has a capacity for 160 beds for women. We work under the guidance of the Women and Child Development. And anybody earning 10,000 to 50,000 can be accommodated in this hostel. Very economically priced and safe haven for any girl trying to stand on her own feet and be financially independent in a city like Mumbai. A healthcare center through which we have several activities. We reach out in camps and also provide facilities of a physiotherapist and a dentist on campus. Computer center, we reach out to little ones who want to learn new techniques on the computer, as well as senior citizens who still have to figure out how to use the computer to reach out to their young ones. And that's how we have these. And we also have very important programs in bioinformatics, which are used through these computers. A library supported by the Maharashtra state government, which provides reading room facilities, as well as around 6,000 books within the library. A scholarship awards program, which is actually funded with the well-wishers who give us money donations. The interest from the same go out as awards to young girls who have done meritoriously in the 10th, the 12th standard, BSc, MSc, and PhD program. Our most recent award session took place about 2 weeks ago. We had Dr. Vasudas Kamath, who was the previous Vice Chancellor of SNDT University, speaking to the girls on the new education policy. And many of the young girls were awarded, around 20 of them received awards from these donation interests. We have another program, which is a vocational program, which is early childhood care and education program. I will tell you a little bit later in my next slide. The daycare center and the nursery school, which are much looked for by the working women, unfortunately has to be temporarily closed during the pandemic days. We look forward to opening it as soon as permission is granted. As I mentioned, this early childhood care and education program is for young girls, and age is also not any bar. Those who have completed 12th standard, they can join it, and it is affiliated to the SNDT Women's University. After completing the course, they can be teachers in any daycare and nursery schools. In addition, we have parallel programs for shadow training, teachers training, and for understanding learning deficiencies, which is done along with Drishti, another NGO. And so young girls who are looking for these kind of programs do connect with us on our website or send us a mail. In the past year, we have been acknowledged by the Vigyan Prasar, and through them we have reached out to pre-primary and primary teachers, and our early care and education teachers have made a program to teach the teachers how to teach young ones. And this was a program well appreciated by all those who participated. Our computer education center completed 30 years, and announced that we had a seminar on changing face of the digital era. On the science education initiatives, we have lots of programs. One of them is for the school students, the other for college students and faculty. For the school students, we have a small laboratory. Here you see one of the volunteers showing an experiment. We have a science nurture initiative where young students from neighboring schools come, and our volunteers help them to understand whatever they are being taught at school in a better way, and they enjoy the science once they've understood the principles, and they leave us very confident young individuals. This program continued online during the pandemic days, and we also have science lectures, science camps. Now these are supported also by Vigyan Prasar. We conduct science exhibitions. So those of you looking to participate, or you know of friends who would like to participate, we will be announcing these shortly for Science Day. For college students and faculty, you are attending one of these popular science lectures right now. We also conduct lecture workshops and refresher courses. The refresher courses are for faculty, while lecture workshops are for college students and faculty, all supported by the science academies. And we have a learning garden where we have around 600 different types of plants. We reach out to community to ensure that they know about these plants, what the attributes, what are their characteristics, what can we get from them. And we have visits. We ourselves go out to exhibitions. We also run some skill development programs through them. In our green initiatives, we have brought solar system for electricity on our campus, and we also have solar water heater for the whole hostel system. Projects in collaboration with Vigyan Prasar are each one teach one. Here, a college student teaches a young student from the school. We are here teaching them to take on the responsibility that they are more privileged than those who need to learn. And through this process, the teacher and the taught enjoy the company of each other, and they learn. One learns to teach, and the other learns from the teacher. And we mentor this process, and this program is helped through Vigyan Prasar. We've had one set with VS College and Somaya. Another one is at the moment going on with these two colleges, where the students are benefiting from these programs. Under Vigyan Prasar, we have a science club, which is referred to as WIPNet. The youngsters have monthly experiments or lectures which bring the youngsters together. Again, through the garden-based, with Vigyan Prasar, we have garden-based workshops on different aspects about how to store seeds, what to do with rose leaves, how to grow mushrooms, and so forth. And these are also exciting programs undertaken by several hundreds of to-be entrepreneurs or even housewives who wish to start such activities in their home. The internship programs for college students we have been doing with Khalsa, SIES, and the other colleges that you see listed here involves our members giving projects to young students from the colleges, and they develop the programs along with expert lectures online, some experiments which they can do in their college or even at their home, and final review by experts on the projects that they have completed. And one such thing is going on right now at this point with SIS College, and we have they are being assessed for what they had done. One of the College, Jain College, actually brought out a little booklet of what they had done, calling the mysteries of the mangrove forest. Their project was on the mangrove projects, forests. We are now with the BRNS supported lectures. This term we have got from 2021 to 24. This set of lectures has been going on since the last 11 years. And we were approaching colleges where we were conducting popular science lectures like this one today. In the last year, we have reached out to nearly 6,000 odd recipients through headquarters and branches, around 60 lectures in different aspects of physics, chemistry, biology, engineering, with eminent speakers from different academic institutes. The last one was held on the 20th, and that was the 22nd lecture. Today we are here on the 23rd lecture which of this year, 2021, which we actually started only in September 21. We have now introduced the school lectures and lectures for society at large. We take lectures in the schools also, and these are being so much appreciated that the schools now approach us to conduct such lectures for their students. For the society at large, we have been conducting programs on health, history, consumerism, lifestyles, and aging, a large variety of topics which would be of interest to all of us who are around. We have a newsletter. I have not been able to tell you all the things that we do. However, they are you will find them in our newsletter, which are present on our website. And so also will you find the programs that are conducted by our 11 IWSA branches. So please do connect with us. You will also find on our website forms to become members of IWSA. Any graduate young girl who wishes to become a member and join us, please do consider seeing our website, getting down the membership form and joining us for it. Our programs are supported through donations and government grants for specific projects. So help us in our endeavors to help others, and we look forward to stay connected with you. You will note that in the presentation I showed that IWSA believes in knowledge sharing, bringing about a change, leading to value creation and promoting innovation with emphasis on science-based methods. With this, I thank you all for your time, and I would like to thank Dr. Dalai for accepting our invitation, and to the college, Smt Kasturbai Walchand College of Arts and Science, for hosting us. We are still in the middle of the year, and we have a whole year of 2020 to go ahead. So I wish all the attendees here today the very best for 2022. I hope the pandemic will not persist for too long. Thank you.
Thank you so much, ma'am, for giving an elaborate information and inspiring us all. Now, moving further, I would like to request Professor Dr. S.P. Nikhalje, sir, to introduce our today's guest of honor. Sir, over to you.
Nikhalji joins already?
Oh, join it. Oh, he joined. He joined.
Yeah. Otherwise, Wadkar sir, you take the charge.
He is there, sir.
Otherwise, Wadkar sir,
Yes, sir.
yeah, he is
Sir.
left. So, Wadkar sir, you have take charge and then introduce her.
तुम्हीच unmute करा.
Introduce him.
होते सर.
वर्कर कंटिन्यू करा.
Worker.
वर्कर.
So he is also leaving. What is happening? I can also do it the same.
These are the technical issues.
These are technical issues. I think Nikaji sir is not having a range. I don't know what is happening. So Worker sir, you introduce him. Or if not
Yes,
possible,
yes,
then-
yes, sir. Within a few seconds, I will.
yeah.
Sorry, Dr. Dalal.
Hello. Sania madam, I
Good morning,
request you to
everyone.
introduce please speaker.
So our today's guest of
निकाल जी सर अनम्यूट होत नाही. हो. चल सर अनम्यूट होत नाही. That's all.
Anushka, what is happening? You are in the department?
No, sir, I'm not.
Dr. Nikhaj is leaving and again joining. Maybe a range issue.
If not possible, then
नाही ना आपल्याकडे काय नाही. आणि मॉडल सांग.
Maybe a range issue, sir.
If you permit me, I could introduce Dr. Dalal and we can move on with the.
Yeah. No.
Yeah.
Actually, Worker sir will introduce him. If not possible, then it is our duty to introduce him. Yeah.
Yes, please do.
Yeah. Worker sir, what is happening? So shall I introduce?
Yes.
Good morning, all of you. Today is our guest of honor, Dr.
काय झालं अडचण? So Worker sir, is there any range problem with you also?
So sir, it's now.
Okay. Ask him to and immediately start it. Ask her.
Yes, sir, I am here.
Yeah. Sania, you start immediately.
Yes, sir. So today is our guest of honor, Dr. Saurabh Dalal, sir. He is a scientific officer at ACTRAT, Tata Memorial Center. He has completed his BSc from St. Xavier College, Mumbai University, in the field of study life sciences and biochemistry. His PhD study has been conducted in the Stackle School of Graduate Biomedical Science, Tufts University, Boston. He has completed his postdoc in the Dana-Farber Cancer Institute, Harvard Medical School, in the field of cancer cell biology. After that, he is currently working as a PI at Sierra Abstract Tata Memorial Center, and same he is studying. His work is on the cancer cell biology. The research interest of his work is the role of 43 proteins in a regulation cellular pathways and identification of mechanisms regulating therapy resistant in collateral cancer, awards and honors he has given as the Research Fellow of Leukemia Society of America from July 1997 to July 2000, and also he is a Joint Secretary Indian Association of Cancer Research from April 2009 to March 2012. He is also a Secretary Indian Association of Cancer Research from April 2012 to 2014. Also, he has working as an editor Journal of Biosciences from 2011 to 2021. Currently Science and a PLOS one of from 2018. He is also a member of the task forces on cancer cell biology from 2012 to 2014. He has published around 38 publications, including two book chapters. Also, he has some patents in his name. The patent for our transgenic mouse protocol entitled Novel Process for generation of transgenic animals with overexpression or knockdown of different gene products recombinant lentivirus to infect spermatogenal stem cells in retrubant animal. He has also uh, he has also a patent in the antibodies against lipocanil 2 and use through of. Also, he has worked for antibodies against lipocanin 2 use of their and uses of their. So he has granted two projects, one DBT, one DAS, one DAE, SRC, Outstanding Scientist Award. So so we got this. So we welcome Dr. So we welcome Dr.
इसमें
Saurabh.
का होत नहीं
So we welcome Dr. Saurabh Dalal sir for guiding us and representing his webinar topic of the today, the applications of recombinant DNA technology in science and medicine. Over to you, sir. Thank you.
Thank you so much. Thank you so much, ma'am. Without further ado, I would like to request Dr. MD Vadmare sir to take charge of the most awaited webinar. Over to you, sir.
Thank you. Thank you, Anushka. At the very outset, I'm very much thankful to college for giving opportunity to chair this auspicious event in the presence of Dr. Saurabh Dalal and Dr. Sureka Jingde Madam, auspicious and learned personalities in their respective fields. So, without wasting time, I request Dr. Saurabh Dalal to go for his speech, please. Dr. Saurabh Dalal.
Okay.
Yes, sir.
Hi, can you all see my slides?
Yes, sir.
Yes, sir.
yes
Okay.
Actually,
Good.
yeah.
Yes.
All right.
Full screen.
So
Make it
yeah,
full screen.
it is full screen. It is full screen. So first, I'd like to thank the Kasturba College for having me as a speaker, and I'd like to thank Dr. Latkankar, the principal, and Dr. Wadkar, who helped coordinate this event, and all the other teachers and heads of department at Kasturba College. And of course, the students for being here on a Saturday. I'd like to also thank Sureka for organizing this, and IWSA and PRNS for funding the program. So what I'm going to do today is I'm going to talk about the application of recombinant DNA technology in science and medicine. And what I thought I would do is I would first sort of focus on the principles that we think of when we're talking about recombinant DNA technology. It's a field that has been in existence for, I don't know, ever since the 60s. And it has helped us make great strides both in science and in medicine. And what I want to do first is cover the basics of recombinant DNA technology, and then I will illustrate with examples both from science and medicine to see how this is applied. Okay. So before I start talking about recombinant DNA technology, I want to introduce what most of you know as the central dogma of molecular biology. Okay. Now, most of you know that our genome is basically a double-stranded DNA, or many molecules of double-stranded DNA. Okay. And this, the information encoded in DNA is converted by transcription to RNA, a single-stranded RNA. Generally, mostly we think of mRNA, though there are obviously other types of RNAs available. But for the purposes of today, we'll focus on messenger RNA, which can then be translated using these machines called ribosomes into a functional protein. And the proteins, for the most part, are these are the engines that drive biology in many ways and allow cells to function. Now, while this is the central dogma and it is mostly true, many of you are probably aware that certain viruses actually start out with an RNA genome, and through a process called reverse transcription, okay, synthesize DNA from RNA, okay, and I mentioned this process here because it's going to be really because the discovery of the enzyme that did this reverse transcriptase really revolutionized molecular biology and biotechnology and took it and allowed us to take it forward to places where it hadn't been. So, and I will talk about that later in the talk. So, what is recombinant DNA technology? Very basically, recombinant DNA technology is introducing DNA from another species, another cell type, or another individual into another species, cell type, or individual. Okay. So what I'm going to show you on the next slide are the basic processes by which, okay, we do this. So at first, what we do is we have what's known as the donor DNA. That is the DNA you want to introduce either into another a species or another organism. We have things that we call vector.
Just check if the others are seeing the.
Sure. Can everybody see the slides?
No, sir. It is only first blank side is open there.
Okay. Sorry. Let me.
I think you might want to reload it.
Yeah. Let me do that.
F5. So press F5. Yeah.
Okay. Can you?
Yes, sir. It is getting.
Can you guys see the slides now?
Yes, sir. Yes.
Yeah.
It is.
Yes.
Okay. I'm going to stop presenting and start presenting again. Okay. Just give me a second. Okay. I will start sharing again. I know what has happened here. So can you see the slides?
Yes, sir.
No.
Yes.
Okay, I'm moving the slides now. Can you see the slides move?
No, sir.
Okay. All right.
So try to press F5 button after sharing.
Okay, you just give me a second. Let me get out of here. Out of the show. Okay. You can see the slides now, right?
Yeah. Yes, sir. Now press
Okay.
F5, sir.
Yeah, I have already pressed F5.
Okay.
Can you see the slides move?
No, sir.
Okay. I'm gonna try. I'm gonna try this. Okay. I'm gonna try doing a screen share. Can you see my screen now?
Yes. Yes, sir.
You should be able to see my screen. Can you see my
Yes.
screen?
Yes.
Okay. All right. Now I'm going to do this. And now can you see the slides move?
Yes,
No,
sir.
sir.
Your first slide is now there. Title slide is there.
It's not
Yes.
moving, but yeah,
Yes.
we can see
Now,
the movement.
Now it's moving, right?
Yes,
Yes.
sir.
Yes, sir.
All right. So good. So this is the slide I was on. So of course, first you have the donor DNA, which has the gene that you want to clone. And then you have vectors. And the most commonly used vectors we have are plasmid vectors. And I will talk about this in subsequent slides. Okay. So these the donor DNA and the vector DNA is digested with something that we call a restriction enzyme. And I will talk about all of these individually. So I'll come back to that. And then we and then they are these two fragments are incubated together in the presence of an enzyme called the DNA ligase, which attaches these two molecules together. This DNA is then transformed into bacterial cells, mostly E. coli, and then you grow up and then you select for the E. coli that carries the plasmid, and then you can amplify the E. coli just by growing them in the presence of the appropriate media and identify lots and lots of plasmid and then do further experiments with it. Okay. So what I'm going to do next is talk about plasmids. Okay. The first thing so a plasmid is an extra chromosomal. That means it's not part of the genome, genetic element, and most plasmids are actually circular. Okay. And they're double-stranded DNA. All plasmids that we use, generally we grow them in bacteria. So they have, the first thing they need to have is a bacterial origin of replication. So that the plasmid can replicate in bacteria, and then it can, when the bacteria divide, the plasmid is segregated to the two different daughter cells, thereby resulting in amplification of the plasmid. Now, the bacteria will not retain the plasmid unless you give the bacteria a reason to retain the plasmid. So what generally is done is these plasmids have an antibiotic resistance gene. And so after introducing the plasmid into the bacterium, you plate it on media that contains the antibiotic. So only bacteria that have the plasmid will grow. Okay? Right? Because this is an additional burden for the bacterium. So the bacterium is not going to make plasmid for you if it doesn't help the bacterium survive in some way. Once you have this, the third thing, one of the other things that a plasmid needs to have is a promoter for gene expression. Now, if you want to make and restriction sites where you can incorporate the gene that you are interested in. These restriction sites generally are in what is known as a multiple cloning site, where you have nowadays plasmids where multiple sites for different restriction enzymes are sort of placed in tandem. So you can cut using the restriction enzymes that work for you and put in your inserted gene. The nature of the promoter depends on what you want to do next. If you want to express the protein encoded by this gene in bacteria, you need a bacterial promoter. if you want to introduce this gene into eukaryotic cells, and today mostly I'm going to be talking about mammalian cells, okay, you have to have a mammalian promoter. Okay? There are some places where you can actually have hybrid promoters, where you have a combined mammalian promoter sequences and bacterial promoter sequences that allow you to express the protein both in bacteria and in mammalian cells. Finally, if you want to go into mammalian cells, you need another selectable marker. Okay, this is also often an antibiotic resistance gene, such as neomycin or puromycin that confers resistance to either neomycin or puromycin or zeocin or any of a number of other antibiotics that kill mammalian cells. This allows you to stably introduce this plasmid into a mammalian cell, thereby you now have the ability of taking this mammalian cell and studying what happens when you have expressed this protein in this mammalian cell. Okay? So now there are multiple types of restriction enzymes, but for today, I'm going to focus on the most commonly used restriction enzyme in molecular biology, that are the type 2 restriction enzymes. So type 2 restriction enzymes cleave DNA at a sequence that is palindromic. What do I mean by that? If this is the 5' end and this is the 3' end, on one strand you can see that the sequence that it cuts at is GAA-TTC, and this is the other strand. You can see that the sequence reads the same on the other strand. So this is a palindrome, and what this specific type 2 restriction enzyme EcoR1 does is it cuts here and it cuts here, okay? Thus creating a 5' overhang here and a 3' overhang here. What this does, okay, now because these sequences are identical, if you have two different DNA fragments cut with the same enzyme, you can mix them. They will anneal over here, and then because of this annealing, because the sequences are identical, you can then join them together using DNA ligase. Okay, and this is how T4 DNA ligase works. This is the most common enzyme that is used for ligation. Okay, now T4 DNA ligase has certain requirements. It needs the 5' phosphate and a 3' hydroxyl because it catalyzes formation of this this bond, the phosphodiester bond. Okay, it needs ATP as a source of energy, and it needs magnesium as a cofactor. Okay, so what happens is when you incubate these two DNA fragments that have overlapping sticky ends, in the presence of ligase and ATP and magnesium chloride, these sticky ends will first anneal to one another. Ligase will come and bind and then catalyze the formation of the phosphodiester bond, leading to fusion of the two different DNA fragments. Okay. Now, often what will happen is that when you're introducing a gene into a multiple cloning site, you will cut the gene at the 5' end with one restriction enzyme and at the 3' end with another restriction enzyme. Okay, these create different sticky ends, right? And what that means is that if you've cut is that you can only go into the gene that you want to insert only goes into the plasmid in one direction. This is unidirectional cloning, and it's important because since transcription will start from the promoter, okay, the protein will be the gene will be translated, transcribed and translated appropriately. Because if it went in the other orientation, you would actually not get synthesis of the desired gene. What happens, unfortunately, is sometimes we don't have the luxury, okay, of using two different restriction enzymes. Okay, so what do you do then? And this is a problem, and I'll tell you why this is a problem. So if you cut a plasmid with one restriction enzyme, clearly there will be sticky ends when you cut it. And when you add ligase to this reaction, okay, and say you have an insert cut with the same enzyme, when you do the ligation reaction, the intramolecular ligation, that means between two ends of the same molecule, is orders of magnitude more efficient than intermolecular ligation. Okay? Because of that, what you effectively end up getting is you will just get the original plasmid back, which is not what you want. You want a recombinant clone. So the way around this is, remember I said that the T4DNA ligase needs a 5' phosphate for it to be able to form a phosphodiester bond. What you do is you take your plasmid vector and treat it with a phosphatase, either calf intestinal phosphatase or shrimp alkaline phosphatase. There are many phosphatases available now. What this does is it removes the phosphate, okay? Thereby the only phosphate present is on your insert. And then when you do the ligation, okay, only this combination will actually undergo a successful ligation, which means that you will get the clone, the recombinant clone you need, and not the original plasmid. What you might also observe or have figured out is that when you do it this way, you can get it either in the right, what we call the right orientation, in which the gene will be synthesized, or in the wrong orientation, in which the gene will not be synthesized. Okay, but you can get around this. You can get around this by performing additional restriction digests to confirm that the plasmid you have isolated has the insert in the correct orientation. Okay, once you do that, you now have a recombinant plasmid that you can use for experiments, or if you're working in industry, you can use this now for production of the proteins that you're interested in. Now, I'm sure most of you have heard about the polymerase chain reaction, but I thought I would go through it because this is one of these discoveries that, while being a mostly technical discovery, has so enhanced recombinant DNA technology that the person who actually did the first PCR got a Nobel Prize for it. Okay? And this works on a very simple principle. The principle is that if you have a DNA template, okay, but you want to make multiple copies of it, and it's hard sometimes to make multiple copies of a DNA that you're interested in, what you do simply is you take your template, you denature it, and now because of the genome sequencing, we know what the sequence of this DNA is, so we can design oligonucleotide primers that will bind to the 5' and the 3' ends of this gene. Okay? Once these primers, and this is what we call the annealing step. Okay? Once these primers are annealed, you can add DNTPs and you can add a DNA polymerase. And what the DNA polymerase does is it recognizes this primed DNA molecule and then synthesizes new DNA with the help of the DNTPs. Okay, and what this means is that at the end of this first cycle, okay, one molecule of DNA has now become 2 molecules of DNA. Right? You can then keep going until you generate as many copies as you need, and this is our PCR product. Okay. At first, this was done with an E. coli DNA polymerase called the Klenau fragment, which meant that we had to add polymerase after every cycle because denaturation occurs at very high temperatures, as does annealing. An extension for the E. coli DNA polymerase occurred at 37 degrees Celsius. We now have something called thermostable polymerases, which are purified from bacteria that live in thermal vents. And these polymerases are very stable at high temperatures, which means that we don't have to keep adding polymerase at every cycle. We can just add polymerase at the beginning of the cycle and put it into the machine and allow the machine to cycle and get a lot of the PCR product. The PCR allows us to do two things. It allows us to amplify a gene product. It also allows us to modify the gene product so it has a restriction site at either end. You can do that by designing the oligo correctly. So now you have a PCR product that can be digested with the restriction enzyme and introduced into your vector of interest. Okay. What it also does is it allows us to amplify and then sequence the DNA. So, for example, if you were looking for mutations in genes in patients with cancer, for example, you can actually do the amplification and then sequence the PCR product to see if the mutation is present or absent. Now, I'm going to come back to something that I alluded to in the slide where I talked about the central dogma, and that is the process of converting RNA to cDNA. Okay, so you can convert RNA to, so most mRNA in eukaryotic cells, okay, has a poly A tail. Okay, so you can do a couple of things. You can either make a poly DT oligo and then add the enzyme called reverse transcriptase along with DNTPs. And what reverse transcriptase does is it recognizes this this oligo DT primer and now starts adding nucleotides. Okay, and then you have the production of single-stranded DNA, which we call cDNA or complementary DNA. Reverse transcriptase actually gets rid of the RNA strand and then can actually synthesize another strand of DNA, thus getting a double-stranded DNA from RNA. Now, why is this important? Okay, why is this important? This is really important for a couple of reasons. One of the biggest hurdles in cloning mammalian genes and trying to express them in bacteria is that mammalian genes are very large because you have both exons and introns, right? So while a mammalian gene may be many thousands of kilobases, maybe even hundreds of thousands of kilobases in length, the actual mRNA might be maybe 1,000 to 2,000 bases, okay? Now you can clone a 1 kb or a 2 kb fragment into a plasmid, but you can't clone a 100,000 kb, 100K fragment into a plasmid. Okay? So the importance of converting RNA to cDNA is you can actually take RNA from a cell, and if you know the sequence of the RNA, you can make a primer that is specific. It doesn't have to be a poly DT primer, oligo DT primer, and you can synthesize the cDNA using those primers. What this allows you to do, okay, is then take this now 1 or 2 kb fragment, introduce it into the plasmid, and you can make these things in bacteria. Further, what this allows us to do is to generate what we know called cDNA libraries. Okay? For cDNA libraries, you can actually use the oligo DT, or you can use a random nucleotide pool. But most often we use oligo DT. Why are cDNA libraries made? So this is how cDNA libraries are made. You you isolate mRNA from cells, you do the reverse transcription reaction. This double-stranded DNA is then introduced into bacterial plasmids. Plasmids are put into bacteria, you grow, and then you get a bunch of DNA that has different cDNAs cloned into it. What these cDNA libraries allow you to do is they allow you to test they allow you to do a number of different things. One is you can actually use the cDNA library as a source to isolate your gene of interest. You can use it to perform screens, where if you can actually say you have a therapy-resistant cell and you want to convert it into a therapy-sensitive cell, you can put the cDNA plasmids into the cells and see which plasmid actually gives you therapy sensitivity as opposed to resistance, or vice versa. Okay. you can do a lot of different experiments with the cDNA libraries, which is why this process is really essential, and it has actually revolutionized molecular biology for a long time. Okay. So, I've talked about how we can make different clones. So what are the uses of these clones? Okay. In the laboratory, we use these things to generate fusion or recombinant proteins in bacteria. We use this to generate cDNA constructs for expression in mammalian cells. You can now, using mutagenesis protocols, generate mutant cDNA constructs to understand the phenotypes generated by the mutant gene, because biology moves forward by studying the mutant phenotype. And for more recent technology, you can generate knockdown or knockout constructs for studying the consequences of gene loss. Okay. Now, and what I'm going to do now is I'm going to give you a couple of examples for these. Okay. And the first is by generating recombinant proteins in bacteria. Okay. So, why do we generate recombinant proteins in bacteria? You can generate recombinant proteins for a lot of things. Okay. And in this particular example, what we've done is we've taken a gene that we're interested in, and don't worry about the name. The name is not important. Okay. And what we've done is we've fused it to this other gene called GST, okay, to make a recombinant protein that's called GST1433C. And you have GST alone. And what you can do is you can express these proteins in bacteria and purify them on a column. Okay. Now, once you have these purified proteins on a column, you can now ask the question, which cellular proteins does this protein bind to? Okay. So what you do is you make a protein extract from mammalian cells, and you run it over either a GST column or a GST1433 gamma column. Correct? So this is your negative control, because anything that binds to GST, you're not interested in, right? Because it's not something that you're interested in. But only things that bind to this fusion protein are what you're interested in. So once you've done this, you can elute this protein complex. Okay, so we call these protein-protein interactions. You can elute this protein complex from the column and resolve it on an SDS page gel, and then perform a Western blot for different proteins. So what you can see in this slide is that none of the proteins, and this is just the protein extract to show that the protein was present originally in the extract. What you can see is that, okay, the proteins are present in the extract. They are not present when you purify it on the GST alone column, right? This is blank, it's completely blank. Some of these proteins, however, are present on the GST1433 gamma column. So protein 1, protein 2, protein 3, again, the names are not important. I hope that you guys will take the principle with you rather than worrying about names, whereas these two proteins don't. Okay, so from this we could conclude that these three proteins specifically bound to this one. Okay. Now that's great. Now you've done this in vitro biochemistry experiment, right? You said that this protein-protein interaction is important, right? So the next question that you can do by doing these sort of things is ask whether this protein-protein interaction, okay, has an important biological outcome. So what do I mean by that? Okay, all right. So that's on the next slide. Right? So we then took this protein over here and this protein over here, okay, and said, well, these two proteins form a complex. If I make a mutant in this protein that doesn't bind to this protein, okay, what sort of biological phenotype can we see? Okay, so So we made a mutant, okay, that would fail to bind to this. Okay. We did this by changing a single amino acid, a serine at position 236, to an alanine. Okay. What you can see is that both proteins are made. Again, this is the input. The wild type protein binds to 1453 gamma. This mutant protein doesn't form a complex with 1453 gamma. Now, this was interesting because the wild type protein is normally present at the cell border, as you can see in this image over here. This mutant protein that doesn't bind here, okay, does not go to this cell border. Okay. So what this allowed us to do is to conclude that somehow this interaction, okay, between these two proteins was required for the transport of this one to the cell border, okay? And so the protein, if you disrupt this protein-protein interaction, what happens is this protein doesn't go to the cell border, and as a result, what we've demonstrated is that cells that carry this mutant protein don't stick to each other and don't organize to form tissues. Okay. Now this is important because if cells cannot stick to each other to form a three-dimensional tissue, you will not have organs formed. Okay. So protein-protein interactions drive much of biology, and you can understand the biochemical and basis of this protein-protein interaction by using recombinant DNA technology, and then go ahead and see how it affects the biology of the cell. Okay? So that's how recombinant DNA technology helps us study cell biology. So now I'm going to talk about applications in medicine. So remember I said you can use PCR and then sequence the PCR products, okay, to identify potential pathogenic mutations. This is one of the this is an example that's actually in the clinic. And this involves a pair of proteins that are give you a familial disposition to breast and ovarian cancer. That's BRCA1 and BRCA2. Now, work over the years has shown that women who inherit the mutant allele from their parents, okay, are at greater risk for developing breast cancer and ovarian cancer. And there are many pathogenic BRCA1 and BRCA2 mutations that are known and have been established, okay, using family pedigrees as something that would as things that will actually give you tumor progression, or that can predispose women to developing tumors. So when you do this sort of BRCA1 analysis, okay, and if you find the mutation positive, what you can see is that this is associated with an increased cancer risk. Okay. Now, based on this, okay, you can have a clinician can now make a determination as to what advice to give the person. Remember, this woman who has come to the clinic does not actually have cancer, okay? At least does not clearly have detectable breast or ovarian cancer. But based on this mutation data and based on what we know about these mutations, the clinician can then advise these high-risk people, okay, to undergoing to undergo either a clinical breast exam, annual mammograms and MRIs, and in certain extreme cases, drug therapy and preventive surgery. Okay, so this is in the mutation positive patients. In the mutation negative patients, if there is no familial history, okay, and you can just do an annual clinical exam and an annual mammogram. If there is a familial history, you have to do an exam every 6 to 12 months, an annual mammogram and an additional MRI. If you're mutation positive, the risk is much higher, and then you might even actually undergo a risk-reducing surgery or risk-reducing drug therapy. Okay. Now, it's not always that the clinician will recommend a therapy or surgery. This will be done in consultation with the patient after giving the patient all the information that they need to come to an informed conclusion. But it does allow patients to sort of make decisions about their future. Okay? And that's really important. Recombinant proteins. Okay. And this was the first great achievement of recombinant DNA technology in medicine, and that is the production of recombinant insulin. Okay, so you all know what insulin is. Insulin is a hormone, okay, that is required to stimulate glucose utilization. When insulin-based metabolism is defective, you get diabetes, which of course can have quite severe outcomes. In type 1 diabetes or juvenile diabetes, as it is so called, the children do not produce insulin. It's an autoimmune antibody. It's an autoimmune disease. In type 2 diabetes, the cells become insulin. The cells in your pancreas become insulin resistant. That means they don't respond to the normal levels of insulin you produce, which is why you have to give insulin extraneously so that you can lower blood sugar. If you don't lower blood sugar, people get kidney disease, people get heart disease, and this is a huge clinical problem. Okay? Now, in the past, in the very dim and distant past, the only source of insulin was purifying it from pig pancreases. Okay, so you have to get the pig pancreas from the slaughterhouse and purify insulin from it and inject that. What people have done since then, okay, is they have cloned the human insulin-producing gene, made recombinant clones, put them into bacteria, and the bacteria then are grown in a fermentation tank, and this results in the production of insulin. You then purify this insulin by breaking the bacteria open and purifying insulin, okay? And then this pre-pro insulin is cleaved to give you the active insulin, which has two chains, an alpha and a A chain and a B chain that are linked by disulfide bonds. The production of recombinant insulin has greatly improved outcomes for people who have diabetes, for juvenile diabetes and type 2 diabetes. Okay, and without it, we would have many more severe consequences for people with diabetes. The second great example is the production of recombinant antibodies for therapy. Now, you can generate monoclonal antibodies against a protein that you are interested in. You can generate them in mouse. You can nowadays generate them even in the rabbit and in the rat. Okay, and this is a mouse monoclonal antibody, right? And by monoclonal, of course, I mean that it binds to only one epitope. Now, what some of these antibodies do is they will bind to a protein, okay, and inactivate that protein's function. So these are called neutralizing antibodies. And neutralizing antibodies are great because what the neutralizing antibody can do, okay, is it can bind to, say, a gene that, a protein that is driving tumor progression and inactivate that protein's function. Now that's great, and because you can make lots of these murine monoclonal antibodies in tissue culture, and you can scale it up, make it in a fermenter, and purify lots of it. But you can't use these in humans. And the reason you can't use these in humans is because the human immune system will recognize the Fc region of this murine antibody, okay, all right, as foreign, and it will degrade the antibody and you will lose. and so you will not get any therapeutic benefit. So what have people done? They've taken these antibodies, okay? They've cloned the variable regions, both the light chain variable region and the heavy chain variable region. And what they've done is they've taken these variable regions and fused them to the variable, to the constant regions of a human monoclone, okay? These antibodies are called chimeric antibodies. And now when you produce these in cells in culture, okay, what you've done effectively, all right, is now make an antibody that will target the protein you're interested in, but will not be targeted by the host immune system. You can take this one step further, where you can identify only what we call the complementary determining regions in the variable regions. That is the regions that are actually required for complex formation with the protein that you are targeting. You can put these regions into a human antibody in the right positions. And now you have a very humanized antibody that has even greater efficiency at targeting the directed proteins in patients. Okay? There are several of these now in the clinics. You have Herceptin, which targets something called HER2/neu, which is a growth factor receptor that is often amplified in breast cancer. Okay, you have antibodies targeting vascular endothelial growth factor, which are these again upregulated in many tumor types. Okay, now I've shown you recombinant antibodies and insulin are just two different examples, okay, of how recombinant DNA technology is used in medicine. There are many other examples: human growth hormone, erythropoietin, clotting factors. All of these are now used to treat patients with conditions where these things are absent. Okay. I'm now going to move on to a more recent technology, which is real-time PCR. Okay. Now, so what is real-time PCR? Real-time PCR allows you to is a polymerase chain reaction that allows you to monitor formation of the PCR product in real time. So how does this work? There are two basic ways of doing it. I'll talk about both. The first is a fluorescent dye-based real-time PCR. Okay, so you have a double-stranded DNA template. Okay, you denature it, and when you do the PCR reaction, you have this fluorophore present in the reaction mix. Now, this fluorophore, when it is free and not bound to DNA, it fluoresces but not a lot. So we call it a ground state fluorophore. Okay, but when it is bound to DNA and integrated, it's excited. Okay, that means that it will fluoresce much brighter. Okay? So what happens is, as the double-strand DNA forms, you will get increasing amounts of this fluorophore binding to the double-stranded DNA, and you will get increased fluorescence. Okay? And this fluorescence can be monitored. Okay? And in the next slide, I will explain how monitoring this fluorescence gives you a real-time picture of how the PCR reaction is proceeding. The other way that they do it is by using a DNA probe that has a fluorophore at one end and what we call a quencher at the other end. So the presence of the quencher suppresses the fluorescence from the fluorescent probe. Okay? When you denature this DNA probe will bind somewhere in the center of the molecule, okay, not where the primers are needed. And as the primers are extended, the polymerase used for PCR, Taq polymerase, has an exonuclease like most of the DNA polymerases. So what it will do as it moves along is it will cleave, it will digest this DNA probe over here. thereby separating the fluorophore from the quencher and allowing the fluorescence to happen. Okay. So how does this allow you to monitor it in real time? So what you have here is that as the PCR is progressing, depending on how, now what you can notice from here is depending on how much product is made, you will have more excited fluorophore. Okay, because you will have, this will be in excess, this will be in excess. So therefore, only the presence of the excited fluorophore will increase as the amount of DNA increases. Now, your reaction mix has a threshold level of fluorescence, okay, which you determine right at the beginning when there are low cycles. So this threshold line is there. Anything below this threshold line means you haven't generated a PCR product. Okay. Once the fluorescence crosses this threshold line, that's what we call a CT value. Okay. This CT value, the lower the CT value, that means the more the original template you had in the mixture. Okay, because you need fewer cycles. Okay, to get a detectable fluorescence signal. And this will be linear, and then as the amount of fluorophore in the mixture drops, it will become, you will enter a plateau phase. So now, what do we use the real-time PCR for? In the lab, we actually use it to study mRNA levels. Okay, and this is an example from our lab. Again, the names are not important. Okay, so what we've done here is we've purified mRNA from normal tissue and tumor tissue, okay, converted it to cDNA, and then done a real-time PCR analysis for 2 different genes. Okay, and again, the names of these genes are not important. What you can see is that both of these genes are expressed at much higher levels in the tumor, okay, than in the normal tissue. So this tells us that the fact that it is being enriched in the tumor as compared to the normal tissue suggests, okay, that these genes might be somehow driving tumor progression. Right? Not only that, what we can do is we can take the levels of mRNA of gene A and the levels of mRNA in gene B and plot one of them on the y-axis and one of them on the x-axis and perform what we call a correlation study. And what you can see is that when the expression of both genes is you get a linear line suggesting that as the expression of that these genes, the expression of the 2 genes goes up simultaneously in these tumor samples. Okay, and this is what we call a correlation coefficient. Okay, and it's the p-value that you would get this. The probability of getting this correlation due to random chance is very, 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 very low. Okay. What this sort of experiment tells us in the laboratory is that it is very likely that these 2 genes actually function as part of the same cellular pathway that drives tumor progression. And then because we have this data from primary human tumors, we can go back to our in vitro assays in the laboratory and perform experiments to show that these 2 actually are on the same path. So this is how real-time PCR actually guides us in the laboratory. Real-time PCR, of course, as probably most of you are aware, is also used in diagnostics, and I think that the greatest example given where we are at this point is, of course, the detection of COVID-19, right? I'm sure some of you had an RT-PCR test for COVID-19. If you haven't, you're lucky because it's a painful process. But basically what people do is they take a nasopharyngeal or oropharyngeal swab and they store it in a solution that will prevent the viral RNA from being degraded. Okay, the virus is then inactivated and the RNA is extracted. It is then converted to cDNA, and you then do a real-time PCR. And what's done for COVID-19 is that a certain CT value has been assigned as if it is above the CT value, it is negative, and if it is below the CT value, the person is positive. And that is exactly what you will see, okay, in your different COVID-19 report from the pathology laboratory. The other thing that is done that real-time PCR can do for diagnostics comes from cancer. I had said earlier that there is a gene that is often overproduced in cancer called HER2. It's a growth factor receptor. There is an antibody that targets HER2. Okay? Now, the problem is not every breast cancer has HER2 amplification. And these antibody-based therapies are expensive. So you only want, okay, to give this therapy to a patient where the tumor has a HER2 amplification. There is no point in giving this very expensive treatment to a patient that does not have HER2 amplification. And the way people do it is you can do it in two ways. One is the mRNA level of HER2 can go up. So you can convert the RNA to cDNA and do a real-time PCR. Or sometimes this HER2 gene is amplified. That means you have many copies of HER2 DNA in the cell. And then you can just purify genomic DNA and do a real-time PCR for HER2. And what you can see is that you can have clearly this person over here has very high levels of HER2, either mRNA or DNA. This person has slightly less, but still high. This person has even less. And this person probably has a normal component of HER2. Okay? So in these patients, it's probably the high HER2 that is driving tumor progression. And you can take these patients and give them the antibody therapy for HER2. And most likely, the antibody and that results in a regression of the tumor and better outcomes for the patient. Okay? So this is my last slide. I'm going to stop now and thank all of you for your attention. And I'm happy to take any questions that you might have. Thank you all so much for joining me.
Thank you, Dr. Saurabh Dalal, sir, for such a fantastic and interesting lecture. We all are really fascinated about the applications of recombinant DNA technology. You gave us new direction to our thought process. It was indeed a very informative session. Surely our students will be greatly benefited from your talk. Thank you so much, sir.
Sure.
Now for the interaction session. Thank you, Dr. Vadmare, sir. Now for the further interaction session, I would like to call Dr. Professor M. Yadav ma'am as the chairperson. Please, ma'am, over to you.
Thank you. Now section is open for interaction. Participant, if you want to ask some doubts, questions, you can directly ask. You unmute and you can ask your doubts or questions or any discussion. Students.
Excuse me, sir, I have a doubt.
Yeah.
Okay.
Sir, in the recent times, as there are many vaccines that are produced for coronavirus, etc. So, how beneficial are our DNA vaccines over the other processes that are made?
I mean, the data on this is sort of mixed. So the recombinant DNA vaccines are mostly generated with recombinant adenoviruses. So you basically clone the spike protein of the coronavirus into an adenoviral vector and make a recombinant adenovirus. What the recombinant adenovirus then is, when it's injected into you, this is Covishield, for example. Okay? When it's injected into you, what it does is it gets into your cells. You get production of the adenovirus of the spike protein, which then elicits an immune response. Okay. The other type of vaccine that is commonly used now is the RNA vaccine, where basically they package the mRNA for the spike protein in nanoparticle. That's then again introduced, and then that mRNA is translated, and you make spike protein. So far, from at least the available data, the mRNA vaccines seem to have slightly greater protection. Okay, but again, they will not stop infection. None of these will stop infection, but they will prevent severe disease. Okay? You also now in the market, sooner or later, you will have recombinant protein-based vaccines, where you inject a mixture of proteins from the coronavirus, again packaged in like a nanoparticle, and that will again elicit an immune response, and those are also coming into the market soon. That's basically what Covaxin is. Covaxin is basically inactivated virus. Okay, what the advantage of the recombinant protein vaccines is that you can do 2 or 3 different viral proteins, not just the spike protein. Okay, because there are some reports that even the nucleoprotein from the virus elicits an antibody response in human beings. Okay, so you are getting a sort of a wider range of antibodies being produced. And of course, this is only one part of the immune response. There are not a lot of studies on what the other arm of the adaptive immune response, that is the T cell response, does. Okay, because those are sort of more involved elements, and it takes better. So I don't know if I've answered your question, but this is basically what the difference between the different vaccines is.
Thank you, sir.
Sir, can I ask one question?
Yeah, go ahead.
There have been cases where the adenoviruses have, created some side effects in the patient. Those who have been vaccinated, you can't call them patients, but those who have been vaccinated, in those the adenoviruses have created some troubles.
Yeah.
I have many friends also who have experienced this. So can you comment on this, please?
So I can only go by the trial data. Okay. I personally know someone who had a bad reaction to the antibody, to the vaccine. So I know that it occurs. But again, but when you're looking at something like a global trial, okay, you have to see whether this happens in a significant number of patients, or test subjects. I will not call them patients, test subjects. Okay, and you have to contrast that with how much protection it gives you against the virus. Okay, and what the initial death rates with the virus are quite high, especially in the second wave when Delta took over. So so it's always in some way a balancing act. Okay, is the benefit from the vaccine greater than the harm from the vaccine? Okay, and statistically speaking, okay, the benefit from the vaccine far outweighs the harm from the vaccine. Okay, and this is something that is true of almost any cancer therapy that we use. Okay, so these are things that are only addressed by large-scale trials. And yes, some people might have issues, and so maybe in a situation where we didn't have this urgent need to get everything back on track and recovering, would have done many more experiments to find out how to screen these individuals out and not give them the vaccine, but that has not happened.
Thank you, sir. That's difficult. But if you have the problems, then you are 100%, sufferer. Yes.
I can only sympathize with people who have those.
Yes.
I mean, as someone who has drug allergies, I had to take the vaccine much later than my other colleagues because I was told we can't give you the vaccine at this point. So, but I had no reaction, so I.
Yeah, even I. So it's okay. Thank you so much, sir.
Yeah.
Hello, sir.
Sir,
can I ask you a question?
Yes, please, go ahead.
Sir, which is the substitute enzyme used in RDN technology as a substituent of restriction enzyme?
Sorry, can you say that again?
Sir, which is the another enzyme used in rDNA technology as a substituent of restriction enzyme?
So, right now we don't, so there are other ways of doing it. So right now in the market, you have kits for making clones that are based on recombination. So there are recombination enzymes, and what you do is you can take you can amplify your gene of interest with PCR and put certain sequences on the end, okay? And then you have a plasmid that has those same sequences. And then you have these recombinases that recognize the sequences and then induce like an homologous recombination, which allows your insert to be be fused with the plasmid. Okay? So there are recombination-based platforms, okay, but these are often available as kits and are quite expensive. So we still use restriction enzymes for almost everything that we do. Okay? But you're right in saying that these sorts of things are available out there.
Yes, sir. Thank you.
Good afternoon, sir.
Yeah.
I want to ask you how nanoparticles are treated. How nanoparticles are used to treat cancer?
So there are a couple of different ways. One is you use something like a gold nanoparticle, which when you shine a light and the particle enters cells, and then when you shine a light on it, it sort of becomes very hot, and that actually results in killing of the cell that it has entered. The other way is to actually take these nanoparticles and use them to package therapeutics. Okay, so what you can do is, one of the great problems that we have with sort of chemotherapeutics, for example, is that you have very bad side effects. They kill normal cells as well, right? What you can do is you can now encapsulate this chemotherapeutic in a nanoparticle that will now only enter tumor cells, for example. And if you do that, what that allows is it allows delivery of the chemotherapeutic specifically to the tumor cell. So it only kills tumor cells and leaves normal cells alone. Okay. So these are some of the things that are being tried right now. I don't know how many of them are actually out there in the clinics, but these are certainly the sorts of approaches that people are trying to do.
Thank you, sir. Sir.
Anybody else?
Hello, sir. Hello.
Yes.
Am I audible? Hello.
Yes.
Yeah.
Anushka, speak.
Sir, I wanted to ask, is there any mutation when we use rDNA technology?
Sorry, can you repeat that question? I lost you for a minute.
Is there any danger of mutation when we use rDNA technology?
Okay, so I'll answer this in a couple of different ways. The first thing is when you're sort of preparing an insert, especially when you're using PCR, okay, there is always a chance that you will introduce a mutation because you have multiple rounds of replication, okay? So in fact, when you do a cloning using a PCR, you always sequence the clone to ensure that the sequence is fine. Okay? Now I suspect that what you are asking about, okay, is introducing a mutation in an organism. Is that correct?
By mistakenly, if
Oh, okay. So here's the thing. You're not. So are we talking about in a mouse, in E. coli, in a human being? Where are we so talking about? So here's the thing. If we just think about human beings, right? There's what we call gene therapy, where we are trying to induce a gene to correct a disorder. So one of the things that, and this has now sort of been taken even further with the use of new technology such as CRISPR, where you can actually modify the genome, right? So there are now guidelines for this. The guideline, first and foremost, okay, is that when you're just doing stuff in the lab, whether you're working on bacteria or whether you're working in cells in culture or even in animals, as long as the biosafety committee clears it, you can go ahead and use it. It's fine because we've been doing these sorts of experiments using other tools. This tool just makes it easier. When it comes to humans, okay, you can correct deficiencies in somatic cells. Okay? So, for example, if you have a tumor that is induced by, say, a mutation in a specific gene forming an oncogene, you can go back in and get rid of the mutant gene using technology, okay? And that should hopefully result in regression of the tumor. Right? Now, again, this has to go through several rounds of trials before it is released. And yes, when you do it in a large enough population, okay, there likely will be some outcomes that we can't foresee that are deleterious. It will happen.
thank you.
So again, there it is risk versus benefit. But you absolutely cannot do germline modifications. So you cannot modify sperm, you cannot modify ova, okay? Or you cannot modify the ovary and the testes. So these are very stringent guidelines. These were guidelines set up by the US National Academy of Sciences, and I believe India has similar guidelines.
Thank you, sir.
Excuse me, sir.
Yeah, maybe I could just add to Saurabh's points. Many of the things that we need to do in human beings have to be cleared at the national level through ethical committees and committees which look after recombinant DNA technologies. So you can't just take any of these things directly to human beings, and that's what he conveyed to you in short.
Yeah, someone else had a question.
Yes, sir. Madhulika Navgare here, sir. Good afternoon.
Yeah, good afternoon.
Sir, I have a doubt. There is a technology known as RISC, RNA-induced silencing mechanism.
Yeah.
So, can it be used somewhere or somehow in human welfare while we are fighting with coronavirus?
So people are trying to do that. People are certainly doing studies in lines and culture, and maybe they've gone to an animal study so far. I don't know, but so far I mean people have been trying to do RNA CRISPR-based RNA diagnostics as well for COVID. I know that I've seen people trying to do that. Whether that's not, at least as far as I know, approach come to widespread usage. But yes, certainly it's possible.
So then why this approach is not utilized, if at all there are so many?
Because you have to be able to get into a cell and do this specifically. And we are still not at the stage, okay, where we can guarantee that, the RISC complex will only degrade the coronavirus RNA and not other RNAs, which might lead to other problems.
Okay, yes, sir. Yes.
Okay?
Yes, sir. Thank you, sir.
Sir, I have one doubt. Does recombinant insulin have any toxicity in human beings?
No, it's been widely used for years now. I mean, it's like I said, it is the poster child for recombinant DNA technology. Okay? In fact, if you ask anybody from a biotechnology program, what's a great example of biotechnology in medicine, the first thing they say is insulin. Okay, and it is safe. It is not toxic at all. Okay, unless you inject it too much, which means that you've not monitored your blood sugar properly. Okay, so I mean, clearly, if you overdose yourself, that will be an issue. But if you use the right amount, it's perfectly fine.
Yes, thank you.
Good afternoon, sir.
Yeah.
I want to ask a question. Can we use enzymes in antiviral therapy?
Sorry, can you say that again?
So, can we use enzymes in antiviral therapy?
I don't know. I mean, again, I don't know if anybody has tried it. So my reading hasn't extended to this, but I mean, I guess it's possible.
Okay, sir. Thank you.
Sir,
Yeah.
which vaccine is better for COVID?
Sorry?
Which vaccine is better for the COVID? Covaxin or Covishield?
I don't know that one is better than the other. The data says that, as far as I remember, the trial data says that Covishield provides slightly higher degree of protection than Covaxin, but again, that's only what it does is it prevents severe disease. Okay, that means you don't go to hospital, you don't go to, you don't have to be on a ventilator, you don't have to go to the ICU. Okay, unless you have some extremely other sort of complicating medical conditions. Okay, but for most people, getting any vaccine, okay, prevents all these things. In this current Omicron wave that we are going through, we've had quite an outbreak at the institute where I am at. 3 of my students have been infected. But, they've had what looks like a very bad flu, and then one is back to work, the other, 2 of them are back to work, the other one will probably come back to work next week. And they've all had 2 shots. Okay? So the point of the vaccine is not that it will prevent you from being infected, but that you won't get severe life-threatening disease. And, I would encourage all of you life sciences students to please spread this message, okay? Please spread this message. This is an important message, okay, and that needs to go out. And young people like you need to be able to put this message out to people.
Okay, sir. Thank you.
Sir, my another question is that Covishield is recombinant vaccine, but the Sputnik efficacy rate is much more than the Covishield. Why?
So I think one of the reasons is that what Sputnik has done is they've used 2 different adenoviruses. So they package the spike protein in one adenovirus type, and for the second shot, they package it in another adenovirus type. Now, remember, an adenovirus is also a foreign thing, right? So, with Covishield, because it's the same adenovirus, okay, what you might be getting is an immune response to the adenovirus when you have the second shot. Whereas with the Sputnik, because they've used a different adenovirus, okay, for the second shot, you are not going to get a immune response to it. Now, this is theoretically how it might work, okay? I don't know if practically this is true. I don't know what studies they've done, but this is one possible answer.
Okay, thank you, sir. Sir,
Yeah.
sir, as many of us are in BSc Biotechnology, MSc field, so I would like to ask, do you have any advice for us who want to go into research field further?
That's it's $1 million question. So I'm unlike my many of my colleagues, I try to actively discourage students from a career in research. Okay, I'll phrase that differently before Surekha starts yelling at me. Research is difficult.
Hello.
And research is really difficult. It requires two things that are, almost polar opposites in terms of behavioral things. One is that you need to be creative. You need to be able to think in different ways about a problem, novel ways about a problem, based on what is already there, and then move forward into it. Okay. The other problem is that it requires a certain amount of doing the same thing again and again and again. Okay. Because data needs to be reproducible. So you really have to do the same thing again and again and again, multiple times. You have to, confirm an observation with multiple different experimental approaches for you to be able to believe in that observation. Okay. So on the one hand, you have to be very creative, and on the other hand, you have to do this sort of mind-numbing, repetitive work. Okay. So this combination, is really hard for a lot of people to put together. Okay. So this is why research is hard. It's also hard because now I'll only speak about research in biology because I know biology, I don't know other fields, okay? But research in biology is especially difficult because, we're all the products of evolution, right? Now, if you look at things like, a computer, for example, all right, we started with vacuum tubes back in the 1940s, and then, silicon chips and things like that. And, you probably all know what this is, right? This is a pen drive. I think it's a 16 meg pen drive or something like that. It doesn't matter. GB probably now, doesn't matter. The point is that the storage space in this pen drive, not too long ago, would require an entire building, a 4 or 5 story building, to achieve the same storage. Okay? So, with technologies of this sort, what has happened is we've started with the basic stuff, and then we've moved on to sort of more complicated stuff, right? With biology, okay, given human lifespan, whatever, 70, 80 years, okay, and given that mammals like us, okay, for evolution to actually manifest itself takes thousands of years, we have the final product, effectively, in front of us, that we are studying. And this is the result of thousands of years. Maybe hundreds of thousands of years. Okay. So studying biology is extremely difficult, which of course leads to a lot of failures. Okay. So one thing that scientists are extremely used to is rejection and failure. Because our grants get rejected, our papers get rejected, lots of things get rejected. Okay. But the important thing is that when you get something right, when you make a new discovery, no matter how small it is, no matter how small maybe your contribution to the body of knowledge. In my opinion, that's a feeling that's not replicated anywhere. Okay? So if you want to do research and you have the inherent curiosity and the will, yes, please. Okay? Research in biology can be very fulfilling. After a lot of pain and heartache, most times. Okay?
I'd like to add to Saurabh's views. It's very important that you go in for a research career that you wish to enjoy. I think that's an underlying factor. Underlining, write it in big capital letters. Then all the hard work, the repetitiveness does not come in the way. If you do not enjoy the work you're doing, then you will find the many roadblocks, stumbling blocks, and all the challenges which will make you stop. So go in with an open mind, go in ready to enjoy the work, whatever be it, and you will enjoy doing research.
And pick a field that you are interested in.
That's right.
Don't pick something just because it is new and novel and exciting, because it might be new and novel and exciting, then it will become boring 5 years later. Fashions change in science just like they do with your clothes. Okay?
Keep an open mind.
Yeah.
Sir, if you don't mind, I have another question.
Please go ahead.
From your introduction, I read that you have done your PhD from abroad. So can you please elaborate about it? Like after BSc in India, how did you get an opportunity to?
How did I? I did a bachelor's. I did a year of a master's, and then it was a lot. People ask me this, and my thinking is that it's a lot very different now than it was back then. Okay? Because I left India for a PhD in 1989. Most of you weren't born at that time, so that's what those are the timescales we're thinking about. But again, if you're applying to graduate programs, I think there are three things you should keep in mind. Whether here in India or in the US or in Europe or wherever, if you're applying to a department for a PhD, the first most important thing is see if there are people who are doing research that you are interested in that department. And there should be at least 3 to 4 people who are doing science that you are interested in. No? You could, if there's only one person there and that person says, this year I don't want a PhD student, what are you going to do? Are you going to go into a lab where you're not interested in the research? That's not going to work, as Surekha has pointed out. That's the first thing. You need to put in some effort, okay, into thinking about what you want to do a PhD in. And then saying, well, in this department, there are 3 or 4 people who actually do things that I'm interested in. Okay? So that I actually did that back in the days when, we didn't have internet access. So we had to write away to universities for their little booklets,
brochures
that told us who did what. Okay, and then based on that, I applied. Okay, for some reason, then I thought that working on DNA on tumor viruses was important, so I applied to places that had a lot of people who worked on tumor viruses. All right, I've been. Again, if you ask me now, Saurabh, why did you want to do that? I don't have a good answer. And I don't think I had a good answer then, but okay, so that's one. The second is when you write your letters or your statement of purpose, okay, tell them why you want to go to the university or that department. Tell them why you want to go there. Say, this is what interests me. This is what excites me. You have to be able to show people that you understand what they are doing, and that's why you want to go there. Because they get hundreds, thousands of letters. Okay? And then, after that, especially if you go abroad, okay, or even here, make sure that you're compensated for your time, that you have a stipend, okay, and that you're compensated fairly. All right. These things, the amount that you will be paid is important because you have to live on your own. Okay. So make sure that and this information on what PhD students are paid and all now is available on the web. You should ensure that you get what you deserve. Okay.
Yes, sir. So, how was your experience studying abroad, PhD?
Well, I had an excellent time. I was very lucky, and sheer good luck. Okay, this has nothing to do with any intellectual input on my part. Out of the 3 places I got into, I think I ultimately landed up in one of the best teaching departments in all of the US. Okay? There may be departments with more famous scientists, more prominent scientists, but this department had made its goal that it would produce the best possible scientists in it. And this was a departmental goal. It extended across all the professors in that department. And, as someone who runs the graduate, has been part of running the PhD program at ACTREC for many, many years, I've often felt that if I can do a third of the job that they did, I would be doing very well. Okay. So that, and I also was lucky is that in the fact that I went to Tufts, which is in Boston. And the great advantage of Boston is that it's one of the greatest cities to do science anywhere in the world just because of the sheer number of universities that are there. And not just small universities, large universities. Harvard, Tufts, MIT, Boston University, Boston College, Brandeis, okay, and many, many others. And they're affiliated medical schools and all of these things. Okay, so I was exposed to a wide variety of talks, of ideas that, our students here don't get a similar exposure. And these are facts. Okay, we try to get people to sit in talks and listen. But with Boston, I could have spent the entire day, 5 days a week going to talks, and I would have still missed about half of them. So you just had a lot of exposure, and that makes a difference because it's all learning. You may not be studying while you're listening to the talk, but it's all sorts of seeps into your subconsciousness, and you remember it. Okay, so those 2 things I think were what really drove me towards becoming an independent investigator and running my own laboratory.
Yes, sir. Thank you.
I would like to add one more point here, and which is questioned by many youngsters who meet me and who are undergraduates and say, what shall I do next? And I want to do MSc, and where should I go? I think this question is now very easy to follow. All of you should go to all the websites of the institutions. The information there is so exhaustive. And as Saurabh rightly pointed out, understand what they offer, understand what you're interested in, and then make a conscious decision of where you would like to go. But there are umpteen exams that you have to appear for, so that would also help you decide which exams to take up and so forth. Otherwise, you'll be just fumbling as to, what should I do next? And this exercise doesn't start in your 3rd year. of undergraduate, it should start somewhere in your second year. You start, wading through these websites, and you'll find that every time you visit a website, you'll get more and more information, which you collate, and then you shortlist the places that you think are most interesting for you and pursue them, so that you are a very enlightened person by the time you reach your third year and ready to decide your next course of action. That makes you very targeted rather than a lost person at the end of your undergraduate program. So I wish all of you the very best actually in this exercise, and I'm sure you will all do well as you move along.
Yeah. And I'm just going to add one thing to what Surekha said. It's good to have a goal and focus and things like that. But that's not the end. It's just the beginning. Okay. So you might start out by being interested in something else, and then become interested in something else. That's fine. Okay. As long as you do it in a organized, practical fashion, it's perfectly okay. Okay? Science changes. Science is constantly evolving. Our knowledge changes. Okay. Every year we know things that we didn't know the previous year. Okay. So your goals can change, but just make sure you're changing them for the right reasons, and not because something is now fashionable.
Thank you, sir. Thank you, ma'am.
Dr. Watkar, maybe we can.
Yeah. I think most of the
Yes.
questions might have covered. We are.
Sorry, 1 minute. Saurabh can also share his email, and I think the youngsters can connect with him. Is that okay, Saurabh?
Yeah, that's fine. You can find it on the website. It's sdalal@actrec.gov.in, all lowercase.
Maybe in the chat box you can.
I'll put it in the chat box. It's there.
Yeah.
You guys see it? Okay.
Thank you, sir, for clearing all the doubts. And thank you, ma'am, for guiding us in the right manner. Now I request Dr. S.S. Watkar, sir, to orient the thanks.
Thank you, Anushka. Good afternoon to all. As we know, an event is not successful without collaborative efforts of everyone. I'm feeling very privileged to deliver a vote of thanks to those who all have contributed to make this event very successful. On behalf of Smt K.W.College of Arts and Science, Sangli, I extend my deep gratitude to today's speaker, Dr. Dalal, for sparing his valuable time from his busy schedule. And I hope he has opened new windows for these undergraduate students in their career. Thank you very much, sir. I must thank Dr. Surekha Madam, the coordinator and trustee of IWSA, and along with all the trustee members, authorities, and members of the association for offering us to host this webinar. We are fortunate enough for his kindness. Thank you. Thank you very much, madam. I'm very much thankful to our beloved principal, Dr. P.P. Largaonkar, sir, for granting permission to host this webinar. Sir, always motivating us. So keep motivating. I extend my sincere thanks to Head of Departments of Botany, Professor M.D. Vadmare, Head of Department of Zoology, Professor Dr. S.B. Nikhalje, Head Department of Microbiology, Professor Mrs. U.M. Yadav. Thank you very much, sir, for your kind cooperations. I'm grateful to Dr. Jaya Kureka madam. She was the cause for this event. Through her, we can reach with Sureka madam and all this made happen. Thank you very much, madam. I'm also thankful to my all colleagues of the department, non-teaching staff who ensured smooth running of this entire event. I extend my thanks to our computer assistant, Mr. Vardhan Savule and Prashant. Without their support, I think it is not possible to an online platform. I'm also thankful to our final year student Anushka Rai for a very good anchoring. Last but not least, thank you all dear participants who have joined through online platform for listening patiently. Dr. Dalal, I hope this might be a new beginning to you all. Thank you. Thank you, everyone. Thank you, ma'am. Thank you very much.
Thank you.
Okay, thank you, Jindagi madam. Jingde madam, thank you. Sureka madam, thank you.
Thank you, Professor Ladgaonkar. It's been wonderful being with all of you, and I think Saurabh and I both enjoyed the interactions with the youngsters in particular, and we hope all of them benefit from this event here.
Actually, there is one request.
Yes.
Actually, we are having some engineering programs also, MSc Electronics, MSc Embedded Technology, even MSc Computer also. So if such a type of expert lecture available for engineering views, engineering themes, then it will be beneficial for the entire stakeholders of this college. So I'm requesting you, if possible, then please arrange such type of one lecture for the electronics people or the people, those who are having some corporate type of the disciplines.
I'll connect with you separately and then speak on this at a greater length. Through our own connects and so forth, and I'll put you with the right people or be a facilitator for the same. Yeah.
Okay. Yeah.
Yes.
Okay.
We'll do what we can best do from our end. Yeah. Our aim at IWSA is to see that we reach out to all of you wherever you are, and to take science, whether it be engineering or mathematics or biology, whatever be it, to all the institutions, and especially the young ones who would benefit most from the new science and the state of the art, and also what's happening in this country. I think that's more important. They should feel proud of what we are doing.
Yeah.
And I think it's here also. There was a time, like Saurabh said, to consider going out, but I think there's a lot being done here, and we should all be happy about that too, and take the opportunity to enjoy the same and contribute to it. Thank you so much.
Can I put in a word, ma'am?
Yes. Yeah.
I'm very much thankful to you, ma'am, for coordinating such a wonderful lecture. It has been a pleasure listening to Dr. Dalal sir, and to you also, ma'am. And I'm hoping for more such endeavors in future. Let's have a nice collaboration further too, and hoping for a more, educative collaborations with you. Thank you so much.
I'll remain connected with you, Jaya. Thank you.
Thank
I'll remain connected with you.
Just thank you, everybody.
Yeah. Yeah. Thank you, and we'll now disconnect if you permit.
Yes.
Yeah.
Sure.
Bye.
Thank you. Thank you.
So we will,